All right. Welcome, everybody. I'm just going to let everyone trickle in before officially starting. It's wonderful to see names that I'm starting to recognize. Good to see you all, or to at least know that you're there on the other side. And we are recording. Okay, great. So welcome everybody to this, our first webinar for semester 2. And I'm Nicola Elliott-Wong. I'm the program coordinator at H3D Foundation. And today's talk, entitled Open Source AI for Infectious Disease Research, Lessons Learned from the Field, is by Dr. Gemma Turon, CEO and co-founder of Acilia Open Source Initiative. Before handing over to her, I'd just like to remind everyone to please stay muted. There will be a Q&A at the end, and feel free to post questions in the chat as we go along. So a little bit about Gemma. Trained as a molecular biologist, Gemma completed a PhD in colorectal cancer and stem cells at the Institute for Research in Biomedicine in Barcelona in 2019, before taking a one-year break to focus on working and volunteering in the third sector. This shifted her scientific interest to global health and neglected diseases and the existing barriers to tackling some of the most urgent health issues in developing countries. With Acilia, she aims to explore new ways of community building and engagement in the scientific arena at the intersection between academia, biotech startups, and NPOs. So I'm working with Gemma now quite a lot, and it's let me tell you that they are very busy. Gemma, I'd like to say if you need a 5-minute nap halfway through this presentation, I would support that from a health perspective. But we are so chuffed to have you opening our semester talks for this semester. So chuffed to have you. Thank you so much for doing it. And I'm going to hand over to you now. Over to you, Gemma.
Thanks, Nicola, and thanks for the introduction. Yes, it's great to be working together with the H3D Foundation, and also great to see some recognizable names here in the audience. So I hope it's not too boring what I have to share with you today. So we are gonna chat a bit about our approach to develop open source AI tools to support infectious disease research across the Global South. And we are gonna try to build it up in how our tools have evolved as we have been learning what was people's needs, basically by working with several organizations, including H3D. So I hope you enjoy this journey with me. So Acilia is a tech nonprofit. We work, our vision is to reduce the existing inequalities in global health, and we basically equip laboratories across the Global South in low and middle income countries with AI tools to support infectious disease research. Why do we work in this space as scientists? Acilia was founded by myself, Miquel, and Eduardo. We were three, mostly coming from an academic background as doctors. And what we realized is that the disease areas that we were focused were mostly non-communicable diseases, which are a important health problem, but there was still a whole dearth of infectious diseases, as you know well, that were not being tackled. Just to put some graphics into that, this is the world. If we plot the countries according to their land area, colored by its World Bank classification by income, and what happens if instead of plotting the land area, we do plot the infectious disease burden, we see how Africa and in general low and lower middle income countries harbor most of this infectious disease burden. But on the contrary, if we plot the publications as a measure of the scientific output from these countries, unfortunately, and as we all probably know well in this audience, these countries are still not the major producers of science in the world, which still is retained by North America and Europe. And of course, this has direct implications on the health issues that are being most actively researched. To put some number to these inequalities, 6 of the top 10 causes of death in low-income countries are still due to infections, but at the same time, those countries produce less than 5% of the world's research, which leads to a number of drugs not being developed, not being proper number of diseases not being properly studied, with not even 15% of the drugs that are being developed worldwide currently tackling infectious diseases, including major diseases like COVID, malaria, or tuberculosis. So there is really a need to speed up drug discovery for a number of diseases that affect predominantly low and middle income countries. And the best way to do it, of course, is to support researchers in those countries because there is a lot of untapped talent, and there is a need to basically provide a better support for this research to happen. Why do we use AI? Well, we are a small team. Our background expertise has always been AI for drug discovery in non-communicable settings. So we realized that AI basically is a cost-effective manner to do research, is faster to implement than any experimental method, is data-driven, meaning that we can use data that has been developed in other fields and apply it to infectious diseases. In the same way, we can reuse methods that have been already developed elsewhere and for other disease areas and reapply them to infectious diseases. And of course, because it's a growing field, and as scientists, we always like to stay on the top of the developments that are happening. So we focus on bridging this gap between scientific research and infectious diseases using artificial intelligence. So far in these 3 years, we've developed a toolbox that started at the beginning in 2020 by building a platform that would allow us to bring AI tools closer to scientists, basically a way to serve these models in a way that they are easy to use by anyone, hopefully in a no-code manner. We are nearly there. This is called the Ercilia Model Hub, and we currently have around 150 models, but we realized this was not enough, so we also developed automated ways to actually build these models so that non-expert users can also build very good, proficient, good performance AI models. This is the tool that we call Zyracam that we'll also talk about today. And then finally, and especially in light of the most recent developments in artificial intelligence, we've focused on all this pool of generative tools to basically expand the known chemical space for anti-infectives. And we are developing a tool to that end that is called, at the moment, ChemSampler. All right, so let's start by the beginning. Where did we start in 2020? Was the development of this hub of models. So the Ercilia Model Hub idea is that a scientist does not need to know all the steps that go inside training a model. So basically, a lot of time happens here, as some of you already know, in actually getting the raw data and processing it. Once you have this data cleaned up, you need to train a model, validate that this model is working, test it, and hopefully even validate it externally in the laboratory if that's possible. So up to this layer, to this orange level, validation is where most of the academic AI researchers stop at. They publish a paper, and the model sits there in a repository, practically being unused and actually very difficult to access by anyone who is not a computer science expert. So we also focus, what we focus most is in this external layer, what we call the deployment layer, which means that we take all these models, we bundle them, no one needs to see what has happened inside here, but they can simply come with a question, for example, a particular chemical, a particular putative drug, select the right model and get an answer. In this case would be a target predictor, right? So this is what we are trying to build with Ercilia Model Hub. And we, of course, have models from very different sources. We are an open source initiative, so we really aim to promote the reuse of things that have already been developed, that have been published, that are actually very good. So in this example, I'm highlighting here a paper from when we started, actually 2020, which was a seminal paper for antibiotic drug discovery. They developed a very good method called ChemProp. So now this model is incorporated in our hub, so you can simply input the drug, get whether it's active or inactive. We also focus on finding interesting data sets in the literature that are also open source. In this case, this large scale analysis of antimalarials. And we built a model based on that, so that also you can predict whether a particular molecule is active or inactive against malaria using this data. And then finally, and perhaps most importantly, we develop models in collaboration with our partners. I'm highlighting here an example of a project we are working in together with the University of Wea, where we are trying to elucidate natural product activity, right? So where you can basically focus on this natural product space and understand if they have activity against, in this particular project, viruses. Okay, so how does it look as of today? We have, as I said, around 150 models. This is growing. This is being developed in a variety of ways, and it's a big part of what we are building together with the H3D Foundation. So do follow up our LinkedIn's and social media to stay tuned for developments in that area. But before moving on, I want to highlight the importance, as I said, the importance we realize having a community of scientists, and not only scientists, but computer scientists, AI experts, open source enthusiasts around, has helped us build this platform as a small team. So basically, we've built an entire pipeline that is more or less automated with the support of organizations like GitHub, Atlassian, or Harvard University, where there is a public discussion around a specific model. Then a model request is opened. Once this model request is approved, meaning this model fits our requirements, it has to be public, it has to be associated to open source licenses and so on. We create a new repository in GitHub where the model is going to be living. Then these open source contributors work on this model, bring back the code to our main repository in this what we call a pull request. There is a lot of automated checks that happen, a final human check to ensure that everything is working, and then finally this model is released. So this is just to highlight what we have been trying to build, not only as a scientists, but also as a different way of collaborating, or at least improving collaborations between scientists and other disciplines, as could be purely computational scientists and software engineers and so on. Okay, so this is where we were in 2020. This is our background tool, and we've been maintaining and growing it as we go. But we realized this was not enough because there was also the need to be able to train good models based on specific data. So this is the tool that we've called ZyraChem. And now, if you'll allow me, we'll go a bit with a bit of detail into what does it entail to train a model. So the first thing that we need to think about when we train a model is data. Biological data can be organized in many ways. These knowledge graphs summarize basically the interaction between the main entities that we find in biology. So we have compounds, that would be the drugs. These compounds can treat or can cause diseases. These compounds interact with genes, which ultimately define cells, tissues, which pathways their proteins are involved in, and so on and so forth. So basically, this is all the information that you can collect about biological entities. We have, of course, started focusing on the compounds as a drug discovery initiative. So how do we encompass all the knowledge we have about compounds in a way that we can feed those machine learning models with very detailed information? Some of you may be familiar with this kind of concept, which is how do we describe a molecule for a computer? This needs to convert this image that the chemist would understand into something like vectors, numerical vectors that the computer can read. So there is classical descriptors that basically capture information about the connectivity of the molecules, and there are 2D maps, even 3D, and some even now pharmacophores and so on in 4D, let's say. But most interestingly for us, there is also all this new generation of data-driven descriptors, which basically use themselves machine learning tools to generate very accurate representations, and I'm going to show a bit more what we mean by that, of these compounds. So not only about its chemistry, which is basically what the classical descriptors do, but also information about the targets they are involved in, the networks, the cellular effects, and the clinical effects, right? So just to summarize, if you have this compound, as we said, you can basically get information from its chemical structure, but you also have a lot of other information. You have, for example, that it's a known antimalarial, its mode of action, um, whether it has toxicity or not, and so on and so forth. Okay, so all this information we can now encode it in very dense vectors for our machine learning purposes in what we call, in this case, is the chemical checker, which is a method that Mikel, Resilia's co-founder, developed in 2020, and that has been even awarded a Dream Challenge Award at the year it was developed. So basically, just to give you an overview, what we have moved from is from basic information about the chemistry of different disease drugs to a much more comprehensive, which is the chemical checker, description of these compounds. This is important because if we can fit all this information into the actual models, then the models that we produce will be much more accurate. Okay, that said, what usually we encounter, going back to Zyrachem, is a collaborator that has a pool of compounds for which there is known activities. For example, IC50s against a particular pathogen, right? And then we want to basically be able to predict what happens when we have a new compound. Will this be an active or an inactive? So as I said, the first step is featurizing these molecules, converting basically these molecules into these numerical vectors, that the more accurate they are, the better the models will be. And then the second step is fitting a model. So using all these algorithms that will correlate these vectors with the specific output, so that when they see a new vector, they will be able to predict whether it's active or inactive. Here, and just for the purpose of representation, mind you that, of course, this is far from the actual reality. But if you can see the active molecules, the ones in red, which are the first one and the third one, have like a similar vector distribution to this new molecule that we are wanting to know its activity, which probably will mean that the model will predict it's also an active, right? So that's at the core of it, how it works. We just try to map the known actives to a specific feature so that then we can predict with a new molecule whether it will be an active or an inactive. Of course, what happens behind this is actually quite complex. And what our tool is doing, Zyrachem, which was published last year in Nature Communications, is basically focusing on a pool of descriptors. So this is a step of featurization. This includes the chemical checker, but also other kinds of embeddings of 2D fingerprints and so on that I'm not going into detail for the sake of time, but you can check in the paper. So we have this first step of featurization with all these. This is actually quite flexible. So you can, if you have interest in a particular one, you can easily add it to the pipeline, as you can see here. Then, of course, these molecules have to be clean and standardized to make sure that they are all properly processed. And basically, this allows us to get an ensemble of vectors. Each vector comes from a different descriptor. These descriptors are calculated through the Cilia Model Hub, and we get this representation of molecules. Some of the vectors, that's important, they need to be imputed, normalized, a whole bunch of mathematical procedures to ensure everything is fine. But basically, when these procedures are done, we go into the second step, which is the actual model building. And again, here we've opted for a pool of different tools, which include typical, more traditional tree-based classifiers like XGBoost or Random Forest. You may be familiar with these names. We've also looked for what we call transfer learning, which is models that are pre-trained on a very large data set and then are fine-tuned based on your specific data. These are neural networks. We also use dimensionality reduction to try to select the most relevant features. So we use UMAPs and PCA in that pipeline. And then we also use image-based descriptors, where instead of having vectors, you convert to like image type input for the machine learning models. So we do first a pooling of features, then a pooling of different automated methods for training a model to obtain finally a very good performing model. And then hopefully the next slides convince you that our method is actually well performing. But here the key to understand is that all this is automated, so it's just one click of the button away to train a very good model without the user having to decide any of the internal parameters. That is usually also what takes a lot of time and can induce errors during machine learning training. So just to highlight how this tool was developed, this probably you're familiar with this image. It's Table Mountain at the University of Cape Town. So we've partnered with the H3D Center, which is working mostly on anti-malarial and anti-tuberculosis drug discovery. So what happens when we started this collaboration 4 years ago is they had a bunch of internal data on their drug discovery pipelines, but they didn't have AI expertise in-house. So we developed these tools so that they can train these models to mimic a virtual screening cascade, basically mimicking what is happening in the laboratory, but in the computer, so that all the scientists in the center can benefit from this technology and basically speed up their research. So what are the assays that we have modeled? Well, we started with generalistic assays, whole cell assays like IC54 Plasmodium for tuberculosis. We also include cytotoxicity assays, ADME screens like solubility, microsomal clearance, etc., and even some more advanced cytotoxicity and so on, like hERG blockade. So usually, typically in these pipelines, the more advanced you go, the less compounds you have to train models because the attrition rate is quite high in these first stages. So only the best molecules will be tested against these more advanced models. So to supplement that, we've not only used H3D data, but when it has been needed, we've also included external data that we collected from the scientific literature. So now to the point, this is what the output of Zyrachem looks like. This is the pipeline that I just described, where we process the molecules, calculate all its descriptors, do this different pool of estimators, and then finally generate an automated report. So there is a lot of plots here, but let's just focus on one for now, which is basically this. This is giving you the score of the model for molecules that are real inactives and molecules that are real actives. As you can see, the inactives have really low scores and the actives have higher scores. Of course, for some of them, it's difficult to discriminate, but these are actually very good numbers and allow us to very well separate between real actives and inactives, and therefore saving a lot of time before actually doing any experiment in the laboratory. So as I said, we have modeled this whole cascade with all these 15 assays that are written here. For all of them, we have very good performances of areas under the curve are above 0.8, except for some cytochromes, because the cytochromes have very low data. So we have actually improved these numbers by including external data and H3D data at the same time. And basically, these areas under the curve, we have been able to bring them up to over 0.75 with these methods. So just to finalize this section and to showcase how this can be useful, we basically took an example that was already done at H3D. So a whole series that started from this initial hit, okay? And then was expanded to these 65 compounds. And then all 65 compounds had to be synthesized and tested in the laboratory to identify which ones were good ones. So I've highlighted just a few of them. Here on the top, you have the experimental activity. So basically, whether these molecules are active against NF54, Plasmodium falciparum NF54, malaria, Plasmodium falciparum K1, whether they are soluble, they are permeable. And then in blue, we have activities that we want to minimize: cytotoxicity, hERG toxicity, clearance, metabolism by cytochromes, and so on. So basically, what you want to see is a lot of dark red and very little blue. So if you see here, our model, these are the predictions and these are the real values. Our models are actually pretty good in identifying. This was predicted to have this was found to have high activity but be toxic, and our model had already predicted that good activity but toxicity. So basically, this compound wouldn't have been to be synthesized. You could just discard it by its cytotoxic prediction. This compound was looking much better. So it had very good activities and lower toxicity predictions. And indeed, when it was tested in the laboratory, that was the case. This is actually the lead compound for this series. Same case here, good activity, low toxicity predicted, and actually really as well. And this compound, again, good activity, but a lot of toxicity predicted, and indeed experimentally confirmed. So this compound would also not progress forward, right? So this is a way where you can filter out your libraries based on the predictions of models, basically these virtual screening cascades, allowing you to synthesize less molecules and speed up the whole research process, saving time and money. Okay, so I hope this was not too much or too technical. Let's move on to the third part of our toolbox, which is this around expanding the chemical space. So we started by noticing there was a lack of ways to use AI models. We developed the Ercilia Model Hub. We realized there was a a need for training models. We developed Zytachem. And then, especially in the last 2 years, with all this plethora of new generative AI, we realized we could apply this to basically suggest new molecules that could enter pipelines, because all these drug discovery pipelines are quite depleted, especially in front of drug-resistant bacteria and so on. So how does ChemSampler work? Well, instead of having compounds and wanting to predict their activity, what we have now is an activity. And we actually want a model that can give me the compound that will have my desired activity, right? Easy to say, very difficult to do. So I'm just going to highlight our first project in generative AI, which is with the open source malaria consortium that is trying to find patent-free antimalarials. They have a series named Series 4 that they have been progressing for years, but they needed better candidates. So they contacted us and asked if we could contribute with some methods to expand this Series 4. So this is about to appear in press. And basically, we've proposed a pool of candidates. And when tested in the laboratory, we've had over a 50% hit rate, meaning over 50% of the candidates that we've proposed are actually active. I'm not going to go in too much into the details of the technology behind ChemSampler. It's similar to Zytachem. It's automated. It's using a pool of different models to basically propose new compounds. And here we are just highlighting how this works. So we have devised a whole cycle where we start by generating a lot of compounds. So here at the top with a plethora of generative AI models. And then we filter them down based on several activities: desired physicochemical properties, synthetic feasibility. You can include constraints like I just want molecules that can be directly purchased, for example. We want compounds that are not too similar to what we already had. We built some activity predictors, so we also selected compounds that were very actively predicted, and so on. So we kept iterating of our candidates until we arrived at a short list of 35, of which 8 were chosen by the chemists. Just by checking the list of these 35, the 8 that looked best to them were selected to basically undergo experimental testing. Here we are just showing some 2D reduction, 2D dimensionality reductions of all these different stages. What's important is to see how actually, as the more active compounds we filter out, we end up in a very particular region of the chemical space that seems to retain this activity. All right. So with that, we went to the laboratory. We did the first round of synthesis here on the left. We were extremely happy to see, this is the positive control, to see 4 compounds with very, very good activity, especially this one, the number 72, and then a couple with good activities that could be optimized, and then 2 that we discarded for moving forward. And then based on these results, we iterated, we made some changes, and we tried another batch. This other batch also had 4 compounds that look extremely good and 1 that was not bad at all. And then these 4 that we decided to discard. And there is now even a third generation of compounds that we have not included here, but it's also having a similar profile of activities. So basically, this means now open source malaria has all these new lead candidates that they can start optimizing for other desired activities like ADME properties. Perfect. So let me also take a pause here and recap. So we started our journey by focusing a lot on compounds, on drug discovery, serving models, building models using generative AI. But we also started realizing that there were other areas, not purely drug discovery itself, that could benefit from our expertise, especially genomics research. As we all know, African genomes are poorly studied compared to other ethnicities, and therefore AI could bridge the gap while more data is collected in the field. So we have also, in collaboration with H3D, done this project Africa Gradient, focusing on pharmacogenomics research in Africa. If you want to hear more about our perspectives on this, you can read this comment in Nature that we published a bit earlier this year. But so basically, just to summarize this project a bit, and this is yet to be published later. So we have started with the compounds. Now what we want to do is focus on the genes. So the whole pipelines looks as follows. We collect using our knowledge embedding techniques that I have described, like the chemical checker. We collect all the information that we can about drugs, but also all the information that we can about genes. So basically, we have these very dense descriptors of drugs and genes. And what we try to do is simply match pairs of drugs and genes. So if a drug, whether to predict whether a drug and a given gene will have a pharmacogenetic interaction. Yes, we train this model on known pharmacogenetic interactions and try to predict unknown pharmacogenetic interactions. This is step one, okay, machine learning based ranking, where basically we are able to select 50 genes per each drug of interest that are predicted to interact with at a pharmacogenetic level. And then we do a second ranking to select the top 10 compounds using large language models. Large language models are these, for example, GPT. I'm sure most of you have used GPT or a similar interface. So here what we do is we provide very detailed instructions to the large language model. We summarize for it all the information we have about the selected drug and the selected gene, and then we ask the large language model that, based on all its knowledge and all the knowledge that we've passed into it, to basically rank these 50 known, these 50 predicted interactions to select the best 10, okay? And provide an explanation of why, because as you know, language models can sometimes provide misleading data. So we also want to be sure of why is it providing this ranking. This is a step one. So in step one, we obtain predicted pairs of drugs and genes that interact at the pharmacogenetic level. Step two is based on traditional bioinformatics pipelines. We're using, in that case, the available public data on African genomics coming from the 1000 Genomes Project. We identify which genes have abundant and specific genomic variants. So basically, we can classify which genes will be more interesting for to study in the context of African populations. And with all this information, we basically pass our selected genes to the H3D team, which does validate these predictions using PBPK modeling and nonlinear mixed effects models to see if they can understand the variability observed in populations based on those genes. Okay, so just to summarize this part, we've done a lot of work on the identification of abundant and specific variants in ATME genes. Africa has a high variability, so we've defined African abundant variables as those that have over 20% of frequency in African populations, and African specific variants as those that have 8 times more frequency in an African population than any other. All this is defined by the data collected by the 1000 genomes. So we've identified over almost 30 million variants. of which 5% were African abundant and 0.3% were African specific. We've also identified, because this is based on whole genome, that a lot of them are actually intronic variants, so variants that are probably having an effect at the level of regulation, right? Not a direct effect on a mutation, but actually regulation of gene expression. So just to show you the results, we start with having the top 50 predicted genes and drugs. So as I said, we have all these drug embeddings, gene embeddings. We train these classifiers to get these drug gene pairs predictions. So we do all drugs that we are interested in versus all genes. And then we re-rank them with a large language model. This is just a short example of the information that we pass this large language model. And then we ask it to rank the interaction between rifampin in this case and CYP3A4. And then it's explaining us why it's ranked this interaction high. To basically provide this information in a way that it's queryable by its ready researchers, we provide these kind of tables where here we have the list of drugs, in this case, malaria and TB drugs. And as you can see in these dots, what we are trying to highlight is first, whether these genes have somehow been already reported in the literature to interact with the drug. This is the dark dot here, so you can see for isoniazid, which there is a lot of information. Indeed, our model high ranked genes that are known to interact with isoniazid. We've also selected genes that have high scores and that have either African specific variants or abundant variants. So as you can see here, some examples. For bedaquiline, which is a gene for which we know nothing, you can see that none of the genes selected, because it's a drug, and for which we know nothing, none of the genes selected here have actually been reported in the literature, because this is a fairly recent drug. We are able to predict things that make sense. So for example, that cytochrome 3A4 is abranked, but then also cytochrome 3A5 is abranked, because these two genes usually have an effect together on pharmacogenetics. And we also find genes that basically are both African specific, have a lot of African specific and African abundant variants, so they are probably relevant to study in African settings. Okay, so this is how the table looks like. And not only this, but we've also basically created, this is still under development, so we are still working on it. Hopefully this is published soon. But basically, this is a very easy to browse web interface where you can select the chemicals and then you can obtain all the explanations and that we've generated with these large language models. And if you want, you can also obtain the top 50 genes, not only the top 10. All right, so this is what we are focusing now as well, in trying to expand this pharmacogenomics research focusing on African variants. So I think that's been quite a dense presentation. Just to summarize some take-home messages, we are a nonprofit organization. We develop AI tools for drug discovery and development, both at the level of, let's say, drugs, but also looking at genomics and other interesting fields. We are specialized generally in what we call knowledge embedding techniques. This can be for drugs, but as we have seen, this can also be for genes, which is basically being able to obtain very accurate, computer-readable representations of all these biological entities so that we can train very accurate machine learning models. Very importantly, everything we do is free and open source, so you can browse our tools and interact with them and use their outputs for whatever is your research needs. And we are, of course, always open to hearing about interesting data sources, people that want to contribute models to our platforms. All our model contributors are properly attributed. And of course, collaborators that want to continue this work with us. So with that, I want to thank our team, Mikel and Dantry, especially Anna, who did a lot of work on the pharmacogenomics with our board, collaborators at H3D and other centers, and also our different programs that have provided interns and volunteers and our funders. So I think that's it for today. Thank you very much, and I'll take any questions.
Gemma, it's just so cool. It's just so epically cool. So there's a question from Jess. Jess, would you like to unmute yourself and just represent yourself, or would you like me to read aloud? I'm assuming you actually can. Let's check. There we are. There we go. Okay, you should be unmuted now, I hope.
Great. Thank you, Nicola. Yes, this works. Thank you, Gemma. This has really been a great tour through all the work that Acilia is doing. So I was just a bit more curious about the generative modeling work as to whether you're able to incorporate multi-parameter optimization in designing new compounds. For instance, you had the example of activity, but can you incorporate solubility and microsomal stability to generate new compound designs that take into account these properties?
Yes, thanks, Jessica. This is a very good question. And short answer is yes, you can. A bit longer answer is you can do these two ways. So you have one way, which is use a bunch of generative models to basically expand a lot the chemical space and then filter out based on existing models that you have for solubility, microsomal stability. That's, let's say, the large scale modeling approach, which is an option that we've actually applied in this project. But we've also done what we call reinforcement learning, which is at the moment that you are generating the candidates, you have this, that's why it's called reinforcement learning. So you have these learners that basically every time a molecule is generated is scored against parameters that you decide, such as microsomal stability. And then you feed in this information back so that the model keeps learning as it generates the molecules. So you can certainly include this in several ways into your pipelines. The only thing that you need is models that you can trust, right? You need to be able to predict solubility, microsomal stability, or any other parameter that you want. You need to predict these properties in a way that you can trust. That's the only limitation. If you would want to predict an activity for which there is no data at all in the public space or anything, you could not build a model that helps you generate this reinforcement learning, for example. But yeah, hope this answers the question.
Great, thank you. Yes, definitely.
Any other questions? Gemma, if I could ask, if somebody wanted to get more involved with you and with Acilia as a whole, what would be the best way in? I know, for example, you have, younger scientists working with you and that kind of thing. Are there ways in?
Yes. Yeah, thanks for the question, Nicola. So, of course, we have different programs, some of them in collaboration with H3D, of course. So if you are more on the computer engineering part, we do have internships that open quarterly. No, yeah, twice a year, sorry, where we take interns. So that's one way if you're like on the computer science aspect. If you are a scientist, an early career scientist or mid-career scientist with an active research project, the best that I suggest you would do is first follow our courses. So we are giving a course together with HC Foundation soon. So we'll keep offering this regularly. I think that's a great entry point to start working with us. Or if you have a project that you already know or think you know how AI could benefit from it, you can just email me or email Ercilia, and then we'll kind of start discussions into seeing how we could fit this in.
Thank you, Gemma. Olawale, over to you.
Thanks, Gemma. Really, really brilliant work. So I already put my question in the chat, but so the question is if there are small labs across African universities that are doing small-scale drug discovery, but looking at what you just presented, this could be a tool for them to really scale up the work and move faster. But with the goal of eventual commercialization of the discovery work, is there a partnership model that can fit this specific need?
Yes. So if you just want to use our tools without any involvement from Ercilia's side, let's say, that's completely feasible because they are open and our license allows for use for commercial or non-commercial purposes as long as you, of course, credit us for the usage of our tools. If this is a project, let's say that we would develop together, basically because you have data that you would like to model that is more specific to your project. The only IP constraint that we have is that the model, not the data, but if we develop models together, this model will need to end up in our hub, meaning it will be available, the model itself, to other scientists. This is the case of H3D, where the data is not available, but the models that we develop together, they are available so that other scientists can benefit from our work. But the IP of the compounds and of any compounds that could be developed based on these models remains within the laboratory that share their data with us. So that is all. Our only requirement is that the end model that could maybe help other scientists remains open for everyone to be used, but you can keep all the IP for anything you generate and proceed in the best way forward as you may see fit, either for commercial or academic purposes. I hope this helps to understand.
Certainly. Thank you.
Just offer you the mic.
Yeah, thank you very much. I think I've posted my question in there, but basically I wanted to, I think you've answered part of the question already about if I have my own data set from my local lab and I want to train that data so that I can make some predictions on the next round of compounds to make rather than traditionally how I used to make them. Is that possible to use your,
Yes.
your site to train my data for my work?
That is possible, but because of course model training, which would be Zytochem, is a bit more difficult than just making predictions with existing models, I do advise you to contact us if you plan to use our tool or try it out, but at some point do contact us at least so that we can provide some pointers or guidance or just to help out a bit in this space. Unless you have already like a very like an expert in chemoinformatics and machine learning that could look at what models you're generating, I would advise to contact Ercilia to get some feedback on that. Yeah. And I see your other question in the chat about proprietary documentation. So we have an open service for making predictions that at the moment is open. So if you post things there, they are open. You can see them and I can see them and everyone can see them. We are moving everything. You can always download the models and use them locally so that no IP constraints, let's say, are breached. And when we offer our models online in a platform that we are developing, we do not plan on keeping these compounds, but there is always you will submit them to an online server with all that entails. They will not be kept and saved, but they are there temporarily online, right? So I hope this answers.
Bryce, over to you.
Thank you for the presentation, Gemma. So my question is, as far as all drug that shows success is in Maria drug discovery are coming from natural products. What about natural model or natural products?
Yes, that's a great question, and something that, for example, we are actively working with the University of Buea. So natural products, the space is quite unique, as we all know. Some models will perform equally good in the natural product space as they do in the synthetic space, and some others will not. This depends on the data these models have been trained. So whenever the data is available, I do suggest looking at the data and seeing if the compounds are very far, the compounds on which the model was trained are very far from your natural products. If they are not, you can be fairly certain of the let's say, performance of the model. If they are very different, you may want to interpret these results with caution or look for other models. We are incorporating now some interpretability measures in Zyrachem so that you can get a sense of whether the model is doing the right predictions or not. So there is ways to go about it, but as you highlight, if the model has not been trained on natural products, you always need to take the results with a bit of caution, indeed. That is a limitation, or basically that's yeah, how biology works, right? So I don't have a definitive answer. It really depends on the model sometimes. Yeah.
Okay, thank you, Gemma. Thank you. So interesting.
Thanks, Bryce.
Okay, Justice has a follow-up question.
Okay, so thank you for your answer to the previous two questions. I just also want to ask, you mentioned that there are opportunities for internships with you. If someone wants to, I mean, from our group want to come and study and then know what to do in terms of these predictive tools, what background do you require? Because basically, we are all chemists. We are not computational or chemoinformatics or biologists or anything.
That is a very good question. The internships that we have, internships itself within Ercilia, are for computer scientists. This is usually young software engineers that don't have a lot of biology background but have good coding skills and want to get into a more medicine fields and so on. For scientists, there is several routes, like ways to, hopefully a few more in the near future that we are working on with this foundation to set up. At this moment, the best suggestion I have, if you really have people that wants to work with these tools, is first contact us, explain a bit the project that you're working on, and then we can figure out the best way to move this forward, either with a small collaboration and mentoring of, like, for example, a master thesis and things like this. So it really is on a case-by-case basis, though in hopefully not that long into the future, we plan on providing this kind of support more, let's say, a bit more official in like a full program for scientists, not for computer scientists. But this is still under construction, and we are actually actively working on that because we know there is interest. But now I think you need to contact us, explain a bit about yourself and your project, and we see if there is an immediate fit for something that we could do.
I'm going to steal the last question. I'm going to say
Thank you very
5
much.
years, 10 years, 50 years into Ercilia's future. What are your hopes for how these projects and your company, how do they develop? What are you wishing for? What's your wish list?
My wish list is that in 50 years, Ercilia is not needed, right? So we aim to build things that are durable, sustainable, and then that's why they are open, so that in the long term, our mission of bringing basically AI closer to experimental researchers and experimental researchers in areas that have low infrastructure currently are not needed, because there is basically like a data center within each research institution in Africa that can provide, using our tools, or others can provide this expertise to scientists. I'm a wet lab scientist by training, so I don't think every wet lab scientist needs to be a computer scientist. I do think there needs to be good computer scientists that develop things for wet lab scientists so that they can stay where they are doing their work. So yeah, that's my hope, that in 10, 15 years, Ercilia is not needed anymore. So my work is done. I go holidays forever. No, I'm kidding.
You can finally take that nap that we were talking about at the
Exactly.
beginning. Yeah. Okay. Well, thank you, Gemma. Thank you so much for all the work that you do, and thank you especially for giving us this hour. Thank you to everybody who's joined, and especially those who've asked questions. And I can only say, if you're in Cape Town like me, please stay warm. And if you're elsewhere in the world, look after yourselves, and we'll see you back for the next webinar in a couple of weeks. Thank you so much, everybody.
Thank you. Bye-bye.
Thank you.
Bye.
Bye.
Thank you.
Thank you. Bye-bye.
Thank you, Nicola.
Thanks, Justice.
Thank you, Nick. Bye.
Bye, Grace.